said, my name is Sarah Miller. I am a postdoctoral researcher in Kurt Zimmerman's lab at the University of Oklahoma Health Sciences Center. And today I'll be telling you a little bit about Trem2 positive macrophages in cystic kidney disease. So here's my disclaimers. I have no disclosures to make at this time, and then I'm going to skip over my bio because I have quite a few slides I want to get to for you guys. So now, in the cystic kidney, there's a number of different immune cells present. This includes neutrophils, T cells, monocytes, and then the cell type that I'm most interested in for my project, the kidney-resident macrophage, or KRMs. So KRMs are localized kind of adjacent to the cystic epithelial cells right here, and it's long been thought that they promote disease progression. So they are thought to be detrimental in the cystic kidney microenvironment. Now, it's worth noting that the majority of these studies implicating KRMs in the progression of cystic disease and the promotion of disease were done using really rapidly progressing animal models. So the disease time course in these animal models is equivalent to a human developing disease over the span of a couple months for these top two models shown here, or over the span of about 10 years for some of these other models down here lower in the table. And we know that in humans, kidney function is on average normal for the first couple decades of life, with cyst progression usually beginning in the second decade and end-stage disease being reached somewhere between the 5th to 9th decade of life. So this is a really slowly progressing disease, a very gradual process, again, in the average human patient. Now, we wondered, in knowing this and knowing that these rapid models are really not homologous to what we're seeing in human patients, whether the macrophages that are present in these rapid disease models are really going to represent what's occurring in human patients, and whether they might be the same and have the same function and phenotype as macrophages that are present in more slowly progressing animal models, such as the Cre-IFT88 aged or the PKD-RC shown down here at the bottom of the table. So in order to begin asking and addressing this question, which is, are these two macrophages, are the types of macrophages present in these two different model types going to be the same, we utilize the Cre-IFT88 model. And in this mouse model, animals which are Cre positive upon injection of tamoxifen will have this IFT88 gene excised and deleted, leading to the development of cystic disease. And so what we did is we induce our animals with tamoxifen. In one group of mice, we subject them to ischemia-reperfusion injury or a sham surgery as a control. And what this does is this is going to give us a really rapidly progressing cystic disease. So in this group of animals, we end up with our Cre-positive IR group. This is going to be our rapid cystic model group. We have a Cre-positive sham group, which is going to be our rapid model control 1. And then we have a Cre-negative IR group, so another control group, or rapid model control 2. In another group of mice, after tamoxifen injection, we simply wait 7 months and let them age out, and this is going to give us our Cre-positive aged group. So this is our slow cystic model that, again, is more homologous to how disease progresses in humans. And then we also have a Cre-negative aged group, which is going to be our slow model control. So following animal harvest and tissue collection, we sort out which cells we're interested in, and then we send them off for RNA sequencing and analysis. Now, after sequencing and processing our data, we end up with these graphs that are called UMAPs up here. And the UMAPs are showing us the different groups of cells that are present in our data that are termed clusters. And each cluster is going to be shown in a different color. So the clusters are defined by their expression of certain genes. So for instance, if we look at these feature plots down here, we can see we can define clusters 0, 4, and 5 by their expression of C1qa, which tells us that these are going to be kidney-resident macrophage, or again, KRM clusters. Now, after looking at the UMAPs generated for each of our 5 groups, we noticed that we had unique clusters present in both our slow cystic model up here and our rapid cystic model group. So our slow cystic model, we saw an enrichment of cluster 4, which was a Trem2 positive KRM cluster, and we saw that this was significantly enriched when compared with our rapid cystic group as well as our 3 control groups. In contrast, when we look at our rapid cystic model group here, we saw an enrichment in cluster 6, which was a dedifferentiated macrophage cluster. And again, this was significantly enriched in our rapid cystic group when compared with our slow cystic group as well as our 3 control groups. And so our next question is, okay, well, what is the function of these macrophages in these 2 different models? So we began by looking first in our rapidly progressing model. And our lab has actually previously published that when you deplete KRMs in the rapidly progressing cystic model, you actually see an improvement in cystic disease. So what we're looking at here is some H&E stained images of kidneys from the rapidly progressing model. that have either been treated with a macrophage inhibitor, GW2580, or a vehicle control. And you can see that we saw a dramatic improvement in cystic disease in the animals that were treated with the macrophage inhibitor versus those that received the vehicle control. And these findings have actually been corroborated by a number of other groups over the years, all of them showing that macrophage depletion or targeting in these rapidly progressing models significantly reduces cystic disease burden. So this supports the idea that these macrophages, the unique cluster we're seeing in these rapidly progressing animals, are actually promoting cystic disease progression. So now coming back to our slow cystic model and the unique cluster we found there, again, these macrophages were defined by expression of Trem2. They also showed really high expression of the gene MRC1, which encodes the protein CD206. Now this enrichment was found again by single-cell sequencing first, and we wanted to verify these findings by flow cytometry. So unfortunately, Trem2 antibodies don't work great for flow cytometry, so we're defining these macrophages by their expression of CD206 instead. So what we're looking at here are representative flow plots of cells from our various groups. We see F480, which is just a common macrophage marker here on the x-axis, and then CD206 expression is shown on the y. And what we're interested in is this top box here, and this is going to be showing us our CD206 positive or our Trem2 positive macrophages in each of our groups. And we do indeed see an enrichment in these macrophages in our slow cystic group when compared with our rapid group and our 3 control groups here. And then this is quantified in this graph right over here. So we do see an enrichment of these Trem2 positive macrophages in our slow cystic model, both by single cell and by flow cytometry. And this finding was really interesting to us because Trem2 macrophages have actually been found in a number of other slowly progressing diseases. So Ito Amit's group published in 2017 that in the course of Alzheimer's disease in a mouse model, you get the generation of these Trem2 positive macrophages, and they actually serve to restrict the progression of disease. So when they deleted Trem2, they failed to get this Trem2 positive macrophage population coming about, and they saw worsened disease in these animals. So we wondered, are the Trem2 macrophages we're seeing in our slowly progressing disease, are they going to function the same? Like, are they also going to be restricting the disease progression? To begin addressing this, we used our Cre-IFT88 mice, and we crossed them with a Trem2 knockout line. So this gave us 3 different groups here. We're going to have our Cre-positive Trem2 wild-type animals. So these guys are standard, slow-aged cystic model. They have those Trem2 positive macrophages. We have our Cre-positive Trem2 knockout group. So these guys are going to have cystic disease, but they will not have those Trem2 positive macrophages. And then lastly, we have our Cre-negative control group. So after tamoxifen induction, we collect our kidneys and our tissue, and then we analyze it for a variety of cystic disease parameters. So in this slide, I'm showing you kidney weight to body weight ratio, the BUN, cyst count, and then cystic index. And what you'll see is that in all of these parameters, we see an increased cystic disease burden in our Trem2 knockout animals when compared with the wild-type animals. So it does appear that these Trem2 macrophages are indeed restricting disease. So the next question is, how might they be doing this? What is the function that they're having in cystic disease? Well, Trem2 is a surface receptor on cells, so it has a number of different ligands that include lipids, lipoproteins, and various PAMPs and DAMPs. One of the major functions of Trem2, though, is in phagocytosis of apoptotic cells. So we thought potentially Trem2 is functioning as a receptor for these apoptotic cystic epithelial cells and helping to phagocytose them and thus prevent the progression and growth of these cysts in the kidney. So we next used spatial transcriptomic data that we had. So these slides shown here are just from standard Cre-IFT88 aged animals, so they're going to have the Trem2 macrophages. And what we did was develop a signature for Trem2 positive macrophages, which we call cyst-associated macrophages now. So that's going to be this red Trem2 CAM here. We also developed a signature for apoptotic cystic epithelium and then non-apoptotic cystic epithelium. And we projected these signatures back onto our spatial data in order to generate these images here. And this is just kind of showing the localization of these various cell types on our tissue. And what we saw was that near the apoptotic cystic epithelial cells, we had a significant increase in those Trem2 positive cells localized adjacent to those spots when compared with the non-apoptotic cystic epithelial cell spots. Additionally, the areas containing these Trem2 cyst-associated macrophages had an increase in signaling pathways related to cytokine production, extracellular matrix, and then cellular killing. So suggesting that indeed these Trem2 macrophages may be functioning by somehow interacting with the apoptotic cystic epithelium and thereby restricting cyst growth in that way. So we've now found we have a unique macrophage population present in both our slow model and our rapid model. The macrophages in our rapid cystic model appear to be promoting disease progression, while the macrophages present in our slow cystic model appear to be restricting disease progression. But the most important question is, which one of these macrophage clusters is really indicative of what's happening in AD-PKD patients? I mean, which one is more analogous to what's actually going on in the clinic? And so what we did was we used Ben Humphrey's Kidney Interactive Transcriptomics online tool, and we took 2 of the top differentially expressed genes from our dedifferentiated cluster, so our rapid cystic cluster, and then we took 2 of our top differentially expressed genes from our Trem2 cluster, from our slow disease cluster, and we projected these back onto different cell types present in human PKD patients as well as human control kidneys. And the cells we're most interested in, again, are going to be our immune cells down here, so the leukocytes. And intriguingly, what we see is an increase in both the average expression and the percent expression in both of our Trem2 macrophage genes in human PKD patients, indicating that the human PKD patients do have an enriched expression of these Trem2 macrophage genes. So to conclude here, we have found that the phenotype and most likely the function of macrophages in rapidly and slowly progressing cystic disease models are different when we compare them. We've found that their dedifferentiated macrophage cluster that's present in the rapidly progressing disease model seems to be promoting cyst growth, while the Trem2 positive macrophages present in our slowly progressing disease model appear to be restricting growth. We've shown that loss of these Trem2 macrophages worsens cystic disease in our animal model. And lastly, that human AD-PKD patients, or their kidneys at least, have an increase in these Trem2 positive macrophage genes. So with that, I'll just thank everyone in my lab. We have a very large lab now, and it's great, and I love everyone, which is hard to say, I think. Thanks to my funding, especially the PKD Foundation, the research animals which make all of this possible, and then the 2023 PKDCon organizers and participants, and everyone for being here. Thank you.
Thank you so much, Sarah. We do have time for questions. I start right there.
So, thanks so much. Great work. Yeah, but, my question is about the model. You're using ischemia reperfusion injury for the rapid progression. So the question, this ischemia reperfusion injury causing this increase in cluster 6, not the rapid progression itself, the ischemia and the reperfusion you are doing it, causing that.
So that's a good question. I don't know. We don't know that for sure. But I would say these dedifferentiated macrophages have been found in other rapidly progressing models, and then not really related to the dedifferentiated, but as far as the lack of our Trem2s, we've also looked in a couple other rapid models, like the CRE-PKD2s and then the P7s, and I should look and see if they have those dedifferentiated, actually, but they do not have the Trem2s. So I've looked in a number of rapid models.
So
none
it
of
can
them
be
have
due
that.
to, because of the percentage of other clusters is more than Trem2 is diluted, kind of.
So do I think that those are coming about first and
Yeah,
it's kind of
that's why you
like
couldn't,
out-competing them?
Yeah.
That's an interesting idea. I'm not sure. So it seems like Trem2s come about in a lot of different chronic diseases, it seems like. So they're present in obesity and a lot of different cancers, and then obviously Alzheimer's is a big one that they're known for. So it seems like they really develop slowly over time in these chronic diseases. So I'm not sure. Maybe if we let these injured guys go even further until they were.
Anyway, great work. Thank you.
I have a question for you. You think in the end that Trem2 macrophages are good things, right? To have around, that's the idea?
That's the idea.
I'm going with it.
Yeah.
So, two questions or related questions. First question is, how would you drive the other macrophages that are present, good or bad or neutral, into becoming Trem2 macrophages to make this whole idea translational. And then the second question related, you're showing that you have an enrichment of the Trem2 macrophages in your ADPKD patients, which makes sense because they're slowly progressive, so I see that argument. But you have them there, so how do you make them more, or why are they not like doing what they're supposed to be doing or what you're proposing to slow the disease?
And they might be, right? I mean, we don't know the characteristics of the patients.
So how do you make them more then? That's the question.
Good question. So there's actually a Trem2 activating antibody in clinical trials right now for Alzheimer's treatment. It's doing really well, and so we would hope if that makes it through FDA approval, we could kind of co-opt that and use it also in PKD patients as well to promote the activation of these macrophages that are present. And in Alzheimer's, not every microglia is going to be Trem2 positive, right? It's only a portion of the population, but it's still enough to slow the disease progression. And so we would hope that it's something similar in PKD, right? We could use this Trem2 activating antibody to also target that. While it's not majority of the macrophages present, that small population should hopefully still be able to make a difference in disease. So.
Thank you so much. Yep. So with that, we are coming to the last presentation of this session, which is from Michael Berger, who works at the University of Colorado Anschutz Medical Campus.
Alrighty. Can everyone hear me okay? All right, wonderful. All right, so hello. Like you said, my name is Michael Berger. I am a research assistant at the University of Colorado in the laboratory of Dr. Katarina Hopp. So I'm here to present my research today on the impact of acute kidney injury on ADPKD progression and how it seems that that impact is dependent upon the cystic disease burden at the time of injury as well as the immune state of the kidney. So activity disclaimer, you've all seen it before. In terms of disclosures, here they are listed. All right, so we understand that kidney disease dynamics are altered by acute kidney injury. We understand that clinical incidents of acute kidney injury lead to worsening kidney health, and that we understand that they can increase the severity of chronic kidney disease. We also understand that mechanisms of injury repair are dysregulated in cases of ADPKD. So furthermore, we understand that ADPKD patients who experience kidney stones tend to have larger cyst volumes and greater cyst numbers. Overall, they have worse cystic disease. But importantly, in experimental models, we also understand that AKI is known to accelerate the rate of ADPKD progression in inducible mouse models of kidney disease. However, in the literature, this hasn't been established in germline models of kidney disease, nor has it been correlated with the cystic disease state at the time of injury. So that's where the rationale for this study comes, that we were interested in evaluating and correlating the effects of PKD progression from AKI with cystic disease state at the time of injury, as well as given the fact that our laboratory is interested in studying the role of adaptive immunity in ADPKD, we were interested in correlating the effects of AKI on PKD progression with immune state at the time of injury. So we employed 2 mouse models to do so. So the first of which you'll see listed in this presentation in blue throughout the course of this presentation, is the Pax8 PKD1 inducible model of disease. So at 4 weeks, we induced disease with a single doxycycline IP injection, or a series of doxycycline IP injections. And soon after, we induced acute kidney injury with a single IP injection of folic acid. 8 weeks later, we dissected animals and harvested tissues for quantifying disease state. So then the other model that we used in this disease is a well-established preclinical model of ADPKD. It has a germline mutation. At 12 weeks, we induce acute kidney injury, also through a single injection of folic acid. And similarly, 8 weeks later, we dissect the animals and harvest tissues to quantify disease state. So at the time of injury, the severity of the disease between the 2 models is significantly different. That, as you can see on the right side of the screen, here is our inducible model at its baseline, that there are minor dilations or microcysts, but ultimately we functionally consider the kidney to be non-cystic at the time of injury. And that's borne out by, as you can see in these graphs in the blue data, lower baseline kidney weight, body weight, as well as low baseline cystic index. So compare that to what we have here, which is our PKD1 RC animal, is moderately cystic at the time of injury. And these data are also borne out by kidney weight, body weight, and baseline cystic index. So while their disease state is significantly different at the time of injury, what was not different, or there was no significant difference, is the degree to which they receive kidney injury. So as measured by loss in body weight, or elevation in BUN, or relative KIM1 expression as measured through qPCR of whole kidney tissue, there's no significant difference in the degree to which the two different animal models received acute kidney injury. What there was a significant difference in, however, was how they responded to it. So on the left side of the screen, you can see the histology for our inducible model. This animal received acute kidney injury, this animal didn't, and this is 8 weeks following injury. And as you can see, there is a substantial acceleration in the rate of PKD progression in that model, and that is to be expected. It's been demonstrated in the literature before. But what was surprising to us, however, is in the same time period, as you can see on the right side of the screen, we have our PKD1 model. This animal received AKI. This animal did not receive AKI. We didn't see the same degree, or we didn't see an acceleration of PKD progression in response to AKI. And these data are further corroborated by all metrics of disease that we took. So whether it's kidney weight femur length, as you can see in left side in the blue, inducible, right side RC, or endpoint BUN, cystic index, fibrotic index. In every instance, there was a substantial acceleration of disease in the inducible model, but we did not see this acceleration in the RC model. This was contrary to our expectations. So to verify that it wasn't due to the methodology we employed to induce AKI, our collaborator, Dr. Kurt Zimmerman, on the same timeline that we employed for our PKD1 RCRC animals, induced unilateral ischemia reperfusion injury. You can see by the histology listed on the bottom of the screen, this is our IRI kidney, and it had no substantial acceleration when compared to its contralateral kidney or to kidneys that had received a sham surgery. And that's borne out by the same disease metrics as well, the kidney weight, body weight, cystic index. In all instances, there's no significant difference. There's no evidence of an acceleration of disease progression in response to acute kidney injury. So the question then becomes why? We understand that the kidney immune cell profile at the time of injury modulates pathological outcomes of acute kidney injury in non-ADPKD settings. We further understand that cystic kidneys from ADPKD patients present with immune cell infiltration, and that the urine and cystic fluid of patients contains inflammatory signaling models. Experimentally, we understand that immune cells, in mouse models of ADPKD, that immune cells such as macrophages and T cells, they're shown to modulate cystic disease progression in cases of ADPKD. Now, given that our two different mouse models had entirely different cystic disease states at the time of injury, we assumed that there might be a difference in their immune cell composition at the time of injury. And so we went about assessing that. So as you can see here, once again, in green, you have our RC model. In blue, you have our inducible model. In overall immune cells, as measured by CD45, were significantly higher in our PKD1 RC model. And then breaking it down by individual cell types, you see this significantly higher level independent of the cell type assessed. So infiltrating macrophages, resident macrophages, M2-like resident macrophages is measured or denoted by CD206 positivity, as well as T cells. They were all significantly higher in the PKD1 RC kidney at the time of injury. We view this as it having a primed immune system, that it's already experienced a cystic microenvironment, and so therefore there's a more inflammatory immune system at the time of injury, whereas the inducible model has a relatively wild-type immune system at the time of injury. A naive immune system, we like to term it. And these data also are borne out. They're not listed, but they're also borne out with dendritic cells and neutrophils, natural killer T cells. So we were interested beyond just their baseline immune composition. We were interested in what happens in response to injury. That we wanted to see the change in the immune composition in response to injury, and so we went about assessing that by, through flow cytometry, analyzing the immune composition 3 days post-injury. And we saw that there was both animal models received an expansion of their overall immune cells, but the degree to which that expansion happened was significantly greater in the inducible model of disease. And this is even further pronounced dependent on the individual cell type assessed. So infiltrating macrophages experienced an 11-fold expansion in the inducible model versus only a 2-fold expansion in the RC model. In resident macrophages, it's 7-fold versus 2-fold. In T cells, it's 5-fold versus 2-fold. It was especially pronounced in M2-like resident macrophages, which experienced a 54-fold expansion in the inducible model, whereas they only experienced about a 3-fold expansion in the RC model. If you look at, it's not listed, but if you look at M2-like macrophages as a proportion of overall resident macrophages, that increases from about 1.5% to about 40% of all kidney resident macrophages in response to injury. So these are where the descriptive state of our studies lies currently, but they allow us to draw the following conclusions. That we understand that the severity of kidney injury via folic acid is similar between mice that are non-cystic and moderately cystic at the time of injury. But where there is a significant difference is the outcome of PKD progression following this AKI. That inducible PKD1 flox animals, they are non-cystic at the time of injury, but they experience a rapid acceleration in their PKD in response to AKI. Whereas PKD1 RC animals, which are cystic at the time of injury, have no rapid change in their PKD progression. So furthermore, we understand that the kidney immune landscape significantly differs at the time of injury and its response to it. That inducible animals have a naive kidney at the time of injury, but they experience a rapid expansion of all associated immune cell types, whereas PKD1 RC animals with a primed kidney at the time of injury do not experience the same degree of an expansion in their immune cell profile in response to injury. So further directions with this research, we want to expand upon experimental analysis. We want to, or evaluating post-AKI PKD outcomes. We want to conduct longer-term studies on our PKD1 RC animals. We want to injure our inducible model at a time point in which they are already moderately cystic. We also want to assess other modalities of acute kidney injury as well as other potential models of PKD. Furthermore, we want to functionally delineate the role of various immune cell types in AKI response and how it modulates PKD progression. So we want to accomplish this using genetic knockout models. We want to employ adoptive immune cell or bone marrow transfer. Furthermore, we think more broadly, identifying clinical cohorts that allow us to evaluate the impacts of AKI events on PKD progression in ADPKD patients are warranted. In terms of acknowledgements, I'd like to thank the members of the HOP laboratory, as well as our collaborators in the UC Denver PKD program. I'd like to give a special thank you to our collaborator, Kurt Zimmerman, for his help in this. Our funding sources are listed here. And we are accepting postdoctoral or fellow applications, if you know anyone who is interested. So yes, thank you. Is there any questions?
All right, thanks so much, Michael. I will go over there.
Oh, wonderful, very interesting work. Nuria Pastor from the Keck School of Medicine of USC in Los Angeles. So when you did the ischemia reperfusion analysis, did you look at 72 hours time point to see if the degree of injury was the same? In our hands, the outer stripe of the outer medulla looked really damaged at that time point, at 72 hours. And then if we waited longer, we couldn't see much more of a difference between, it was a completely different model, not PKD. So I was wondering if you were planning to look at 72 hours to see how denuded the S3 segment was.
Yes, so with regards to the particular information surrounding the ischemia reperfusion injury, I would like to direct questions to our collaborator, Dr. Kurt Zimmerman. The specific details of when time points were assessed, I couldn't tell you at the present moment.
Yep. And we are doing the bilateral IRA as well to confirm these studies, just to make sure, since this was the unilateral approach. Are there any other questions? Yes.
Very impressive talk. I'm wondering why the folic acid damage is, do the epithelial cells, are they well prepared to the injury, or the inflammatory cells are kind of suppressive. Actually, suppressive function, immune cells in the PKD cysts has a immune suppressive function.
Yes, so not listed in this talk, but one of the things we assessed was the basal level of various immunosuppressive molecules. So we saw that our C kidneys at our C animal at the time of injury, CD8 T cells had a higher positivity rate for PD-1 expression, as well as there was a higher CD25 positivity rate amongst our CD4 T cells, so a higher degree of Tregs in the kidney at the time of injury. And that's one of the things that we're considering might lead to the decreased immune response following AKI.
So I guess the thesis here is getting very confusing because they are both long-term models, and I don't know which one to work on now anymore. But the thesis here is that the RC mice don't respond unfavorably to the injury because they're primed, right?
Yes.
Is there a way to unprime them and then see whether they start behaving like the inducible knockouts?
So I can't speak to that specifically. It sounds like an excellent idea to assess going forward.
Is there a way to unprime them?
I'm not sure that there's a way to unprime them. You could obviously try to do it at a much milder disease than where we did, but I think we are thinking more to do it the other way around and getting the PAX model primed and prove that then that is also not leading to an acceleration. I think that is the easier experiment. Well, nothing is easy, but it's the easier experiment at the time being that we're hoping will give us an answer. Any other questions? Jack? That is the last question, so.
Jack Song from USC. Very interesting observation. And then my question is that I know you look at the T cell, you look at the broadly and macrophages. I was just wondering if the specific T cell type or specific macrophage that what the previous Sarah was talk about the slow progression correlate with the specific macrophage or T cell type that could happen that enriching your RC model, but not in the other PKD model.
Yes, so overall macrophage populations expanded about at the same rate that overall immune cell populations did in most instances, like whether it's infiltrating macrophages or resident macrophages in M1 or M2 phenotype. Where we did see a significant difference and where we think there's something worth further investigation is regards to the not in the RC animal, but in the inducible model, in response to injury, there was a significant expansion of M2-like macrophages. So that there's further delineation that's going to be necessary with this project to determine mechanistically which immune cell we believe to be directly responsible for the difference in responses. But the one that had the most substantial difference between the two models of disease, specifically regarding macrophages, was M2-like resident macrophages. There was a much more considerable expansion in inducible models than RC models.